In the mid of December 2019, a group of patients presented to ER of different hospitals of Wuhan, China. They had following presenting complaints. They had fever and malaise, they had dry cough, and they developed shortness of breath. All these features, due to all these features, they were diagnosed as cases of atypical pneumonia. Now, what is atypical pneumonia? Of course, atypical pneumonia is that pneumonia which is not typical, meaning thereby that it is not caused by that organism that usually cause pneumonia. It is due to some atypical causative agent. Plus, its feature is not that of a typical pneumonia. Its clinical features are different from clinical features of typical pneumonia. Typically, pneumonia presents with high-grade fever, chills, productive cough, and of course, shortness of breath. But here, there is low-grade fever and malaise. Along with that, there is not productive cough, but dry cough. And along with that, of course, there is shortness of breath. And the cause is not that typical organism, that strep pneumonia and other organisms that typically cause pneumonia. So the diagnosis of this is atypical pneumonia. Now what happened that some of these patients, they went on to develop respiratory distress. They developed respiratory distress and they needed ventilatory support even. And unfortunately, some of the patients even died due to this disease. One thing to notice is that there is a seafood market in China, in Wuhan. There is a city called Huanan, and in Huanan there is Huanan Seafood Market. Now, what the doctors noticed on detailed inquiry, what they noticed is that all these patients of atypical pneumonia, either these patients, they were working in this Huanan Seafood Market or they had recently visited this area. So what doctors realized is that this Huanan Seafood Market, this is actually the focus of infection. It is the point source of infection. They said that there is something wrong with this market, that is source of infection is Huanan Seafood Market. Now when they did the bronchoscopy of these patients and they did bronchoalveolar lavage. What is bronchoalveolar lavage is that you pass bronchoscope into their lungs, and from alveoli and bronchioles, they bring this fluid, they bring the alveolar fluid, and when they checked this alveolar fluid under microscope and certain techniques such as PCR, they found a supposedly new virus in their bronchial fluid. They found this new virus and they labeled this new virus as 2019 novel coronavirus. Novel means new. So 2019 novel coronavirus, short form of which is 2019 nCoV. And of course, because this story started in Wuhan, so they also started informally calling it as Wuhan coronavirus. So today we will discuss about this novel coronavirus. Hello, I am Dr. Azaz from MedicoVisual.com. Welcome to this visual lecture. Now what they noticed is that when they did a genomic study of this supposedly new coronavirus, they found out that it resembles with that of SARS coronavirus. There is a coronavirus called SARS coronavirus. They found out that its genome is 70% similar to that SARS coronavirus. SARS coronavirus is a coronavirus that caused an outbreak of severe acute respiratory syndrome in 2002 and 2003 in China. So they found out that it is similar to that. But later on, they actually found out that this supposedly new virus, which they were calling as 2019 novel coronavirus, doctor realized that this new coronavirus is not new. It is actually a strain of SARS coronavirus. It is a strain of SARS coronavirus. So they named it SARS2 coronavirus. So it was revealed that both SARS coronavirus, which previously caused outbreak, and this supposedly new coronavirus, that is SARS2 coronavirus, both of these coronaviruses, SARS1 and SARS2, both of these, they belong to same species of coronaviruses, and this same species of coronavirus is called SARS-R coronavirus. R's mean related, so SARS related coronaviruses, right? So SARS mean, of course, as I told you, it means severe acute respiratory syndrome. Severe acute respiratory syndrome is a type of atypical pneumonia, or you can say it is a type of respiratory illness that is caused by SARS coronavirus. And then there is COVID-19. COVID-19 is coronavirus disease of 2019. It is the atypical pneumonia, it is the disease, it is the respiratory illness that is caused by SARS-2 coronavirus. So the disease that is caused by SARS-2 coronavirus, it is called as COVID-19, and WHO suggested this name. So official name of this disease is COVID-19, right? Now another thing, these coronaviruses, these are not new. We have known coronavirus for some decades. So previously we know that many of these species of coronavirus, they caused common cold. So these are quite common, these are not new. So they were not thought to be much dangerous. But nonetheless, some of these species, they have been known to cause serious outbreak in the human history, serious diseases outbreaks in the human histories. For example, as I told you, SARS outbreak was there in China in 2002 to 2003. Then there was MERS outbreak. MERS is Middle Eastern Respiratory Syndrome. It is similar to SARS, but because the outbreak started in Middle East, so they named it Middle Eastern Respiratory Syndrome, and MERS was caused by MERS coronavirus. Then of course there is COVID-19 outbreak in 2019 and 2020. Now we are going to discuss some characteristic features of coronaviruses. So we will discuss some characteristic features of coronaviruses. All these coronaviruses, they belong to a family of viruses, and this family is called Coronaviridae. They belong to family of virus called Coronaviridae. So first we will talk about its genome. These are not DNA viruses, these are RNA virus. So genome, as far as genome is concerned, they are RNA viruses. And their RNA is single-stranded RNA, and their RNA is not segmented, it is continuous. Of course, you know some viruses, their genome is segmented, but the RNA, the genome of coronavirus is continuous and non-segmented. Then their RNA strand is positive sense RNA strand. Now what do I mean by positive sense RNA strand? Okay, let's talk about positive sense RNA strand. Let's suppose this is the mRNA. So this is the mRNA, and from this mRNA, of course, you know, proteins can be synthesized. So proteins can directly be translated from mRNA. mRNA can be directly translated into proteins. But let's imagine this genomic sequence. Let's suppose this is the sequence of RNA, let's say present in coronavirus. Now if you look carefully, this genomic sequence, this sequence of nucleotides in genome of this virus, it is exactly same as that of mRNA. So sequence is exactly same as that of mRNA. So if the genomic sequence, nucleotide sequence of genome is exactly same as mRNA, it is called positive sense RNA. It is called positive sense RNA. And because it is exactly similar, exactly same as mRNA, it can be directly translated into proteins. So proteins can be directly translated from positive strand RNA. So positive strand, positive sense RNA, it can be directly used as mRNA. It can be directly used as mRNA. Now let's imagine another sequence. Now look at this. Rather than A there is U, rather than U there is A, rather than G there is C, and so on. What you see here is that this sequence, this particular sequence of nucleotide, it is exactly opposite to this. It is exactly opposite, or you can say it is complementary to mRNA. It is complementary to mRNA. Complementary means that if these strands they come close, they can bind with each other like this because their nucleotides are opposite to each other, they are complementary. So such sequence is negative sense RNA sequence. This is negative sense RNA strand, and to make sense out of this negative sense, I mean to make proteins out of this negative sense RNA, it must first be converted into positive sense, meaning thereby that a copy of it should be made, a complementary copy of it should be made, and that complementary copy will act as mRNA, and with that can be used as messenger RNA. So can this be directly translated into proteins? Of course not. It cannot be directly translated into protein. So this was positive sense RNA. Another feature of genome of coronaviruses is that they form nested subgenomic mRNA. Now what do I mean by nested subgenomic mRNA? I'm not gonna explain this right now. For that you have to wait for the next part. In that part we will discuss replicative cycle of coronaviruses family, and in that these things will be clear. But for now you just remember that they form nested subgenomic RNA, and because of this property, taxonomically they are arranged in order of nidovirales. So order is nidovirales and family is coronaviridae. So this was about genome of coronaviruses. Now let's talk about capsid. What is capsid? The genome of any virus, and specifically coronavirus here, it is closely linked, or it is attached with a proteinaceous coat. It is attached with proteinaceous coat, and that proteinaceous coat is called capsid. So proteinaceous coat of virus is capsid. Capsid impart a particular shape to virus, and the shape of capsid is helical. So there is helical capsid in coronavirus. This helical capsid is further arranged in a spherical manner inside the envelope. We will discuss about envelope right now, but right now you understand that the capsid is helical in shape, which lies spherically inside the envelope of virus. Now if you look carefully, I have zoomed into the picture. You can see that this capsid, it is made up of these small subunits. So these small subunits are basically proteinaceous subunits. So these proteinaceous subunit that forms the capsid of virus, these are called capsomers. So capsid is formed by proteinaceous subunit capsomers, and they arrange in a particular fashion. They attaches with each other in a particular fashion which imparts a specific shape to virus. Because of this particular arrangement of these capsomers, the shape of coronavirus capsid is helical. And along with that, of course, inside the capsomer you can see a little bit shadow of RNA. So capsid along with RNA, capsid plus RNA, if we consider them as a single entity, that is called nucleocapsid. Here it should be capsid. So capsid plus RNA is what? Nucleocapsid. So this was nucleocapsid, and you know nucleocapsid is helical in shape. Now we will discuss about its envelope. Not all viruses have an envelope. Those viruses which have envelope, they are called enveloped virus. Enveloped viruses, and of course coronavirus is enveloped virus. Now enveloped viruses, where do they get this envelope? Actually they steal this envelope from the host. Let's suppose this is the envelope and it is actually stolen from the host. How come? Okay, let's imagine this scenario. Okay, let's say this is a virus, and when this virus it exits the cell, it takes a part of cell membrane of the host. I mean, when it exits the host, this virus, when it exits the host, it takes a part of its cell membrane along with it. Look, so it pinches off, and now this part of cell membrane of the host, it becomes envelope of that virus. Most of the enveloped viruses, they get their envelope from the host cell membrane, host plasma membrane. But there are some viruses which for some reason do not like cell membrane, so they get the envelope from nuclear membrane. Okay, like this. So some viruses, they get envelope from nuclear membrane of the host, for example herpes virus. Then there are certain other viruses which gets their cell membrane, which gets their viral envelope from endoplasmic reticulum of the host. So from endoplasmic reticulum of the host, coronavirus is one of those viruses which gets its envelope from endoplasmic reticulum of the host. It gets its envelope from endoplasmic reticulum of the host, which it infects. Now when it steals the host membrane and turn it into its own envelope, what it does is that it customizes its envelope. It decorates the envelope with its own viral protein. It embeds its own viral proteins into this envelope. For example, here you can see there are M protein or membrane proteins that are embedded by coronavirus into this envelope. Then there are E proteins or envelope proteins. M proteins are membrane proteins, E proteins are envelope proteins. These are the viral proteins of coronavirus which are embedded into its envelope, right? So the details of these proteins are not quite well understood, so I will not be discussing them into details. But there is an other viral protein which is most important for this virus, and that is these spiky proteins. These spiky proteins are spike proteins. These are called spike protein or S proteins. These are most important for the coronaviruses. Why? Because these are host receptor binding proteins. These are what? Host receptor binding proteins. What they do is that with the help of these S proteins, these viruses, they bind with the host receptors. They bind with the host receptor and they infect those cells. They bind with certain host receptor, they enter into those cells and then they infect those cells with these receptor binding proteins. And it is said that these receptor binding proteins, they impart tissue tropism and host tropism to the virus. And what I mean by tissue tropism and host tropism is that certain any pathogen, irrespective of virus or bacteria, it can infect certain specific it can infect only a certain specific tissues. It can only infect certain specific tissue or tissues, and this predilection towards certain specific tissues is called tissue tropism. And there is host tropism, meaning thereby that it can only infect, it can only bind with certain specific hosts receptors and it can only infect that host only. It can infect certain specific host. For example, a virus of a cat, let's say, it may not be able to infect human because cat's proteins, cat's receptor would be different from that of humans. So because of these host binding proteins, they have certain specific shape which can only bind certain specific receptors of the host. For example, there is this receptor and then there is another this receptor. Of course, you can understand by looking at it that this spike proteins, it can only bind with this protein, this receptors, and it will be activated and it will enter into cell and infect the cell. It cannot bind with this receptor. Let's suppose this receptor with which it is able to bind, this is actually the receptor present in alveolar epithelium, and this receptor is called ACE2 receptor. So ACE2 receptors, these are present in alveolar epithelium of human lung, and with that it can bind its spike protein and it can recognize it, it will bind its protein and it will enter into cell and it will infect the cell. So because let's say cat would have, let's assume, this kind of ACE receptor and humans have this kind of ACE receptor, so of course it cannot bind with cat's receptors. So because it can only bind with receptors of certain host, that is it has imparted the host tropism to it, the ability to infect certain specific hosts, right? So let's move forward. Okay, here I have shown a more simplified diagram of coronavirus, and this is the envelope and these are the spike proteins. It is much more simpler diagram of coronavirus, and if you look carefully, it looks like a crown of the king when viewed from top. So this is crown of a king. When viewed from top, it resembles coronavirus, or you can say coronavirus resembles crown of the king. Because coronavirus resembles crown, it is called coronavirus. Corona means crown, so that is why it is named as coronavirus. Actually, all the members of Coronaviridae family, they resemble crown because of presence of this characteristically shaped spike proteins. So that's why they are called Coronaviridae. Okay, now interesting thing. Another point I would like to highlight here about enveloped virus versus non-enveloped virus. Of course, you know the coronaviruses, these are enveloped virus. And enveloped virus, because the enveloped is actually derived from host cell membrane, and host is any animal, human or non-human animal. So because we are modern animals, we humans are modern animals, and our membrane, it is not accustomed to harsh environment that was prevalent millions of years back. Right now environment is not that harsh, it is quite favorable. So in that environment our membranes have evolved in such a way that it cannot tolerate very harsh environment. So that's why this membrane of virus, which is actually derived from the host, it can be easily disrupted. It can be easily disrupted by detergents and disinfectants, and with extremes of pH. Even the dry environment for a long duration can disrupt this envelope. Dry environment even can disrupt this envelope. And when this envelope is disrupted, these receptor binding proteins, these spiky proteins, they will fall apart. And when they will fall apart, will this virus be able to recognize and attach the receptor? Well, of course it will not be able to recognize and bind those receptors. Now this is only the virus, this envelope, and of course there will be nucleocapsid inside it. So now it has lost those receptor binding protein. Now it has been rendered harmless. Now it cannot infect any cell, right? Opposite to that, let's imagine naked virus. Of course naked viruses are not nude. By naked I mean these are non-enveloped viruses. So naked viruses, because they don't have envelope, so they have to embed their receptor binding protein into their capsid, right? because they don't have envelope, they have embedded, they have anchored their receptor binding protein into their capsid. But interesting thing is, capsid is the viral protein, and because viruses are primitive organisms, they evolved millions of years back, and when they evolved, they generated kind of their own capsid proteins, and in that environment, when these capsid protein evolved, they are accustomed to that harsh environment that was present millions of years back, and because of it, capsid proteins can tolerate such harsh environment, and because it can tolerate such harsh environment, it is not very easy to disrupt this capsid protein. It is not very easy to denature this capsid protein. So because of this, naked viruses or non-enveloped viruses, these are very difficult to kill. Opposite to that, enveloped viruses are easy to kill, right? So fortunately, our coronaviruses, these are enveloped virus, and that is a good news, because it can be you can prevent coronavirus infection by washing your hands properly with disinfectant and detergents. So you have a question? Okay, so that was the point, that you can easily prevent its infection, right? Okay, so I won't be discussing any more details about enveloped and non-enveloped viruses. We will discuss them when I will start lecture series on basic virology or general virology. So let's move forward. Okay, interesting thing about coronaviruses is that they are thought to be of animal origin. Actually, when scientists they did the genomic study of the coronaviruses, they came to know that their genome, it resembles about 90 to 95% of genome of bat. So they thought that this coronaviruses, they came from bat and they started infecting human. Well, how come? Right now, just told you that there is certain host tropism in viruses, that it can only infect certain specific host. So how a virus from bat jumped into human and start infecting human body? How it can start recognizing the human receptors? How it is possible? Well, actually, that bat virus as such cannot infect humans. But what happens is that what is so unique about viruses is that their genome, they undergo lots and lots of mutation in a short duration, and they keep on accumulating that lots of mutations, and they continue passing these mutation into their daughter cells, into their progeny. And because they continue to accumulate these variations, these mutations, such mutations can lead to so much change in their proteins, that total structure, even the structure of their protein, let's say spike protein, their structure is completely changed. And now this new spike protein, it may recognize a completely new host. Let's suppose in bat the spike proteins was of different type, but when they got so much mutations, the spike protein turns into a new type of protein which started recognizing human receptor. Now the question is that why viruses, they accumulate lots of mutations. Let's assume that this is a viral genome. What happens is that the viruses are primitive subcellular organisms. Their replicative machinery is not that efficient, right? So because their replicative machinery is not that efficient, while replication, sometimes they do mistake. Let's suppose rather than putting U here, it has put A here. So they sometimes do mistakes in replication. They put an other nucleotide than intended into this place. So these things also happen in human cells. In modern organism like humans, I think can also happen. But in humans, what we do is that in humans there is proofreading mechanism, and in human, if such mutations occur, it is quickly rectified by proofreading mechanism, and proofreading mechanism is not that efficient in viral cells. Because of this, they continue to accumulate lots and lots of mutations. Even in human, interesting thing is that if human cells, it failed to rectify the problem. Let's say this is a cell, it has failed to rectify the mutation. What this cell will do is that in such circumstances, it will commit suicide, right? But such things does not happen in viruses. So they accumulate mutations and they start infecting other hosts. So it is thought that from bat, these viruses are actually from their origin is from bats, and then from bats they jumped into an other intermediate host, or maybe they directly jumped into humans, and they started infecting humans. Of course, bat viruses as such did not infect human. They generated new species of virus. They generated some new species of virus due to lots and lots of mutation, and these new species of viruses, these new species of coronaviruses, they started infecting human host, right? Huanan Seafood Market was an excellent place, an ideal place for propagation of these viruses. Why? Because in that market, living and dead animals were put together in very unhygienic conditions. The environment was wet, and of course, enveloped virus, they love wet environment because they cannot tolerate dry environment, right? So in that environment, there was, of course, it was crowdy place. In that crowdy place, there was small streets and environment was wet. In that environment, people were living with animals, and people were living in close contacts with each other and with animals. And in such environment, this virus new strain formed inside another host, and then it jumped into human, and from human, because of close contact through respiratory aerosol droplets, it started infecting other human as well, and then this outbreak of coronavirus happened, unfortunately. Now, this was the discussion of coronavirus, the basic characteristics of coronavirus family. In the next part of this lecture, we will discuss that how this virus replicative cycle of this virus, and of course it replicates in alveolar epithelial cell. But how does, of course, along with that, we will discuss how this virus, how it infects the cell, what is the pathogenesis of COVID-19 or any atypical pneumonia caused by coronavirus. What is its pathophysiology? We will discuss its pathophysiology, its replicative cycle, and then we will discuss its prevention and some aspects of treatment. Treatment is not yet fully discovered, so I won't discussing the details of treatment, and of course I will also discuss points of diagnosis. If you like this video, please subscribe to my channel. Please like this video and give your precious comments. Thank you for watching.